Sorry, I didn't prepare very well. Maybe I can start off by giving some thank yous without going to my PowerPoint thing. In fact, I probably really don't even need it. Well, first of all, thank everyone for coming. This has been a really wonderful day. It made me think maybe I should start a new job so I can retire again, because it was just so much fun. And thank you all. The speakers, that's all been great. And it's been so fun to see former colleagues and so on. And those of you who are online who probably can't hear me now, or maybe you can, I don't know. Thank you for, giving your time to this. It's really been a wonderful occasion. I have a lot of special people to thank. I have some slides here with names on them, but let me just get started. The people who organized this, Ting and Mike White and Tim Shadle and Guyen and Nancy and Kai Tan and Michelle and Eliza all contributed to this happening, and without them it never would have happened. So that I want to thank all of them. Then I wanted to spend a little bit of time talking about going through some history of people who were important to me to get to this point. And of course, first of all, is Larry. It was 52 years ago that I graduated from Caltech and went to Colorado to start graduate school. And he was my second rotation, and I just never left. I found a perfect home. Make one correction, we did play a lot of one-on-one basketball, but that was never our first choice. Our first choice was always to go over to the gym and find two undergrads and take them on 2-on-2. And they were always willing to do it because they were always younger and taller than we were. But I think we won 75% of the time because, like Sean said, I was pretty good at taking people out, and Larry was the Steph Curry of that age, and he could hit the three-pointers from anywhere. So we actually won a lot of games and impressed these young dudes. So Larry, and then one other correction. So Takis mentioned me being in a band. He said it was before I started. It was actually in the middle of grad school. I was in Larry's lab for 3 years. I'd already done my thesis proposal, got it approved, and I just felt like I needed a break. And then I had these two great friends who were musicians and said, why don't you come join us for a band? And so I told Larry I'm going, and he didn't say if you go here or come back. I think he was pretty happy about it, but he let me do it, and fortunately let me come back. But it turned out to be the best possible decision for completely unintended reasons. And that is, so the project that I was going to do, I was going to follow in David Pribnow's footsteps. I was going to isolate RNA and sequence 100 base pairs of RNA that covered a promoter and covered a ribosome binding site. And it was going to take 3 years. I mean, David Pribnow spent 3 years doing it and got a PhD from Harvard, and that was what I was going to do. And so I went away, I came back, and the world had changed. David Pribnow actually came to Larry's lab as a postdoc, but he came from Wally Gilbert's lab where they invented DNA sequencing. And so he did my 3-year project in the course of a couple of months by doing DNA sequencing. So it was done already, which was great. I didn't really look forward to that. You youngsters don't know anything about RNA sequencing. It was truly a laborious task. And I was glad I didn't do that, but that meant I had to find a new project. And so here are the people I want to thank who helped organize this thing. So people who were important to me included, I have to start off with Tom Schneider. So I was looking for a project. DNA sequencing was just barely getting rolling. And I thought, well, what should I sequence? What would be the most interesting things I could sequence to get going? Tom Schneider came as a first-year grad student. He'd come from MIT, and he'd done some programming there. And he took Larry's course. For all the first-year students took his course. And for his term project, he wrote a little program that did some alignments and looked at some things. And Larry showed it to me. I think he was looking at ribosome binding sites. There were about 12 of them at the time or something like that. And Larry showed it to me, and I said, oh, this is really cool. And I went and talked to Tom, and I said, that was great, but what if you did this? Or what if you did that? Or what if you tried this? And Tom gave me the best advice I've ever had. I think he said, why don't you do it? And it took me 2 seconds to say, hey, that's a good idea. So I spent the next few months learning to program. Tom joined Larry's lab, and the rest is history. I mean, we spent another 10 years doing all kinds of stuff together, the perceptron stuff and the information theory stuff, And in fact, experimentally, we also, I think we're one of the first groups to actually utilize randomized DNA to do analysis, selecting for things that would have function. So Tom was instrumental. And then there was a computer scientist who, Andrzej Ehrenfeucht, was the professor of computer science. And we went to him to get help. And he's sort of the one who directed me to perceptrons, which I'd never heard of. And that was a jumping off point. And he was also on Tom's thesis committee on the information theory stuff. But he also had 2 graduate students. And these names will be familiar to most of you. And so for a couple of years, we would get together, 5 of us, every couple of weeks or so, and we would just talk. And we'd tell them, well, tell them about some biology and what would be interesting problems to work on and what you might be able to learn from sequences, DNA sequences or protein sequences. And they would tell us, well, what algorithms might be useful for that. And it was just wonderful. I mean, I learned so much from these 3 guys. And so it was really a wonderful time. A little bit later, George Hartzell came, worked for me, and then Jerry Hertz joined me as a postdoc, and we got into the motif discovery stuff. And that was sort of a jumping off point that sort of made us known to lots of people. Around the same time, we published this paper on motif discovery, and a guy named Alan Lapidus, who was a physicist who actually got his PhD working with Stephen Hawking. But after that, he decided to get into, he was really into machine learning. And so he came back to the US, went to Los Alamos. He was in the complex systems group. And he called me up and said, I liked your motif discovery. That's kind of a machine learning problem. Why don't you come down and we can chat? And I invited my friend John Heumann, who I knew as an undergrad and then as a grad student. And he also had a physics background. And I said, and we had already been talking about some motif ideas and stuff. So I took John with me. And the 3 of us just hit it off really well. And we ended up, he lived in New Mexico and we lived in Colorado. We would get together about once a year and we'd just talk about motifs and protein RNA structure prediction and recognition codes and just all kinds of stuff. And it was just fun, just really an amazing time. I would say the next big event was, and several people have mentioned this, the Aspen Center for Physics, where we managed to get them to invite us to run a workshop on computational biology. The first year, 1990, Christian Burks, Gene Myers, and I were the co-organizers. And that's where I got to know Bill and Steve, and then they became co-organizers later on. They helped organize it. But that first year was amazing in 1990. The heavyweights in the field, Mike Waterman, Temple Smith, David Sankoff were there, and then us sort of mid-career people. And then there were people like Tim Bailey, who was young and hadn't yet gotten into the meme stuff, but he was there. Tim Hunkapiller, Steve Ma. Anyway, it was an amazing collection of people. It was just 25 people. That was the limit. So I love these little small meetings, like somebody mentioned the BAMF meeting, because we could just talk. And it was a 3-week meeting. You go there for 3 weeks in Aspen, which is not a bad place to spend 3 weeks. And we just talk about all kinds of stuff. As Steve mentioned earlier, they were at the time thinking about this block stuff. They hadn't yet published anything on blocks. BLAST hadn't been published yet at this point either, so most people didn't know who Gene Myers was. BLAST wasn't come out for another 6 months or so after that. So anyway, it was just an amazing time, and it was really wonderful. And it kept going for 5 years. I think it actually continued after that, but I wasn't always involved. So I think Sean mentioned Robin Guttell came to Boulder. He had, there was already, one thing I didn't mention about Gene Myers, I meant to earlier. So we were doing all this kind of stuff. Gene actually wrote a program to do minimum free energy prediction of RNA structures. He never published it because Zucker's program paper came out and it was the same thing. He didn't want to just repeat. But I used his program for several different projects after that. Well, so minimum free energy problems was fine, but Robin Guttell was solving RNA structure by comparative analysis, looking for compensatory changes. And he started off as a student with Carl Woese, and then he went to Harry Noller's lab. But they were doing everything by hand. They had small data sets, and they were finding alignments and looking for compensatory changes. So he came to me, and we figured out how to automate what he was doing by hand. And that was a lot of fun. course, the downside of that was you required an alignment. And it took Sean Eddy and sort of simultaneously, but independently, David Hausler to come up with the idea of context-free stochastic context-free grammars, which let you do both the alignment and the structure simultaneously. So that was a big advance that came after that. But it got me interested in RNA, which I had spent a little bit of time with previously. And then Jan Gorodkin, Laurie Heyer, Jack Tabasca, they were looking at RNA motifs. So again, it's a motif problem, but now the secondary structure matters. And it's different than finding an overall fold because you're really looking for a local kind of thing. So that was a bit different. And then Eric Snyder and Sam Levy worked on a different project having to do with gene prediction, which was a very hot topic in those days. Although as soon as people started doing RNA-seq, it didn't matter anymore because you didn't have to predict genes, you could just read them. So that was the point at which I moved here. And these 5 people, so Takis and DeBrage were postdocs who joined me, and DeBrage had a story just like Takis. He wrote to me and said, I'm looking for a postdoc, can I come to your lab? And I said, well, yes, but I'm moving to St. Louis. And he came anyway. And Young Mei and Kai and Ying were all graduate students who are here waiting for me to arrive. They'd already decided they wanted to do computational biology. They were second-year students, so they'd done some rotations and they were waiting for me to come. So what happened was I immediately got here with 5 people in my lab, which is as big as my lab ever was in Boulder. And it let me, get a jump start and get going really quickly. Furthermore, the WashU community as a whole was really welcoming. And that showed up in a couple of different ways. One was, I told you, and I think I've been mentioned, part of the reason I came here was I was convinced that it was time to have computational biology graduate programs. It was clear that sequences were going to come in more and more rapidly, and that people, and there were people doing computational biology, but there wasn't any really training programs in that. And this was a great place for that because there was already a critical mass. Sean was here, Mike Zucker, David States, Warren Gish, Michael Brent had just come here as in the computer science department, and he was changing his direction to work on biological problems. There were people in biochemistry like Jay Ponder, and anyway, there was a great critical mass so that we could put together a curriculum and an admissions committee deciding what kind of students we were looking for. And it got off the ground really quickly. Within a year, we had approval. Next year, we started taking students. It happened kind of slowly. The first year, we only got 2 students, but one of them was Ting. So I'd say that was a successful year. And then within a few years, we had lots and lots of applicants, and it was really took off. And also within the first 10 years, myself and my students had collaborations with over 25 different faculty here at the School of Medicine and 10 different departments. So people wanted us to be here. They wanted to interact. They wanted to use our tools and stuff like that. So it really was a wonderful environment. I forget what's coming next. Oh, so then there was a sort of a next wave of students. Those were the first 3, and then Li Wei Chang and JJ Liu was a postdoc. Ting Wang came here. They were all looking either at motifs or combinations of motifs, looking for regulatory networks. That was the next goal beyond getting to motifs. Look how they go across the genome. Billy Lee talked about his work with Susan. One thing he left out, I should add, is so they did this thing of identifying the ciliary genes because they were in common in humans and clammy and not in plants. And they verified that by basically identifying one gene that was a human gene that scientists had been trying to clone it, and they narrowed it down to about 200 genes. And then Susan just looked at that list and said, it's that one. And she called up the guy at Hopkins who had patients, and he sequenced it, and that was the gene. So they were able to use that to identify a gene, a human disease gene, because it fell in her list of ciliary genes. And within a few years, the whole field of ciliopathies took off based on that kind of thing, because it turned out that lots of human diseases are mutations in ciliary genes. And so now most of her collaborators are actually medical people working on stuff. So anyway. And then more people, Guyan Zhao joined it as a postdoc, Namdi came as a grad student and worked with Jeremy Buehler, who was another computer science faculty member, Aaron Spivak. So these were all people who joined the lab and made it really a wonderful place to be and to talk to. They all had their own projects, but they overlapped enough that we could talk to each other. And around this time is when Scott Wolf contacted me. I guess he said it was Keith first, but I remember talking to Scott early on. From Scott's work with Pebo, they had done a recognition code, which was a deterministic code. That is, for a given protein, you could predict what its preferred binding site was. But everyone knew the whole PWM idea is based on, well, they don't just bind one sequence, they bind a family of sequences that are related to various ways and stuff like that. And so they were collecting huge amounts of data. He told me about his bacterial one hybrid system. I had a small experimental group, but we were doing like one protein at a time and so on. And so when Scott called me up and said he wanted to collaborate, we jumped at the opportunity to have huge amounts of data. And the first person that worked on that was Ryan Christensen, who really pushed that project forward. Excuse me. Later on, Yui Zhao developed better algorithms for analyzing both PBM data and CellX data. Xiusang Rong with Josh Salmidas, another faculty member in biomedical engineering, pushed that whole technique further. And I had a recent student, my last student in computer science, who did sort of deep learning kinds of things to get at motifs. So I had a really wonderful collection of students and postdocs over the years. This isn't all of them. I'm sorry for those of you I've left out, but it was really an amazing group of people. And as I said, I always had a small experimental group, but they did, I think, some really nice stuff starting in Colorado. Dana Fields, then Homsey, Chris Mann, when I first came here, JJ Liu, who did both computational and experimental stuff, motifs, using neural networks actually to model recognition codes. Larry Schriefer worked with me for many years, and David Granis really pushed forward. He also did a combination of experimental and computational stuff and pushed forward the high-throughput CellX. That little logo on here, that's from Zheng Zhao. Did anybody figure out what it is? So that's the lac repressor binding site, where what's shown here is the energy where the average is set to 0. So things above the line have low energy. That is, they're the preferred bases. And this is showing you at a resolution of 0.1 KT, the change in energy for all possible single base pair changes of the lac repressor, which is, of course, the first repressor ever known. And it was a really cool project. The interesting thing about that one was that it binds as a dimer, of course. In fact, it actually binds as a tetramer, with the binding to a single site as a dimer, but it has this asymmetric motif. And that was always a mystery. And it turns out that it has an odd spacing. And if you make it one base pair longer, it becomes symmetric to the right side. And if you make it one base pair shorter, it's symmetric to the left side of the motif. So what happens is you have a homodimer that acts like a heterodimer. It has two modes of binding DNA. And because of the spacing, one binds in the short mode and one binds in the long mode, and you get this really interesting thing. Anyway, that was sort of the culmination of experimental techniques that I've been pushing for 30 years about how you get high accuracy and moderate throughput determination of binding specificity. And then just recently, I've sort of switched to working on post-transcriptional regulation, and had wonderful collaborations with Mike White and Basab Roy and Ishan Hiwa, who are postdocs in the group. So that's sort of the history to get to where we are now. Oh, I had many undergraduates work with me over the years, both in Boulder and here. This one, I'm only showing one. Nanan Zhang was not only a really talented undergraduate researcher, but also a champion Lego designer and maker. And he gave me this as a present when he graduated. Okay, now I have to tell you people who are important to me. And Susan Dutcher, my wife. We've now been together for 40 years. So not quite from the beginning, but almost. He's a great scientist. The only reason that Larry didn't include her on his list of smartest people is he didn't know her very well. He knows her better now. And we love to travel. She's been a great companion and partner. We like going to the same kinds of places and doing the same kinds of things. So that's been wonderful. And besides just a couple of scientific collaborations, we've also had 2 other important collaborations. Children, Ben and Adrian, that picture on the left was taken in China. They're sitting at 15,000 feet, and that peak above them is at 18,000 feet. That was Ben had just graduated high school, and Adrian was just about to start high school. And then the picture on the right, that's with their respective spouses. And now, of course, they have their own families. I can tell you from experience, being a grandfather is great. So anyway, that's it. Thank you all again. It's really been